Here we are. Welcome everybody. Welcome to Cross Stitch with our Cross Stitch a Cell live stream. My name is Alana, but before we begin, let's center ourselves and reflect on the land on which we're about to broadcast from today. We honor the traditional and original owners of this land, the Palawa and Muwinina peoples of Nipaluna, Lutruwita. We pay our respects to elders, past and present, and to the many Aboriginal people that did not make elder status. We fully recognise a history of truth, which acknowledges the impacts of invasion and colonisation upon Aboriginal people, resulting in the forcible removal from their lands. Knowledge makers with deep connection to country, we consider them Australia's first and most important scientists, those who today continue to care for country. Thank you for joining us. We hope you've got a nice cup of tea, you've sat down somewhere comfortable, and you're excited to learn a little bit about some cells and some crafting. So my name is Alana and I am a PhD candidate in the School of Medicine here in Tasmania. My work is in bioinformatics. So that fancy word means that I use maths to study biology, really. So mine focuses on the brain. So neuroscience is something we'll go into a bit of today. And I'm also an avid crafter, but I'm not going to be doing most of the crafting today. That's going to be our artist here with us, Rachel.
Hello, everybody. I'm Rachel. I am a cross-stitch designer by evening, and by day I'm a scientist. I work for the Environment Protection Authority in Tasmania as part of the waste management team.
And I am Shasta. I'm a PhD entomologist, which means that I get to study insects. And I also like craft. I would say that I've gone so far as to actually incorporate insects into the kind of crafting creations that I like to make. This is not one of mine, but I also teach students about how to take scientific information, like how to identify insects, and then cement that information in their brains by then creating insect models. That's one of the insect crafts that I've done. But it has been such a long time since I learned cellular biology. So I am partly here today for Alana to be able to explain cells to me, and you guys can listen in if you want to.
I'll do my best. Well, I think the thing we should start off with first is, what is a cell?
What is a cell?
What is a cell? So we weren't actually aware of cells' existence until the microscope was invented, because they're tiny. They're tiny little building blocks inside your body, each of them their own ecosystem. So back in 1665, when they were first observed, they just kind of looked like little Lego blocks together. But now we have the ability to zoom in and see so much inside a cell. They are their own little ecosystem, and building up together, they're how our body works. They're how most living organisms live. So Rachel has cross-stitched and also Phymoclay, is it?
So pretty.
An amazing cell. The pattern for it as well is in the National Science Week booklet that we have. And you can also find that online if you ever want to follow along. And we are going to do some live cross-stitching today as well. But would you like to tell us first about this amazing creation?
Yeah, so once upon a time, very long ago, I did cell biology as part of my undergrad science, and it was my favourite subject, learning about all the different components of the cell and how tiny they are and how they all have special functions. So yeah, so I tried to replicate as accurately as I can all of the components of a cell in my designs. So this is an animal cell. This is a simplified animal cell. Sorry. And this one is a plant cell, which is slightly different.
What makes them different?
Well, plant cells have interconnections with each other, plasmodesmata, that allow them to communicate with each other. That's the structure.
We have so many funky words for you today.
Yeah, so they can communicate with each other. But yeah, of course, no real animal cell looks exactly like these in real life. They're very specialized and they have particular functions. But this sort of captures all of the basic structure
They're sadly not as colourful as this one in real life either.
No, unless you stain them up with all sorts of fancy chemicals.
It's like an emoji cell.
Yes, that's a great description. This is our emoji cell. So where did we want to start on describing this cell?
Start at the start.
Start at the start. All right. I think the start of a cell would have to be the plasma membrane. That's one of the most important parts, especially for an animal cell, because it's used in several areas of the cell. So around the outside, so we have on Rachel's one coloured in red and yellow, and on the little one, it's purple around the outside there, is this membrane. So think about if you tried to get cling film and form it into a bag and fill it with water. That's kind of like what the membrane is on the cell, but it's not just a solid bag, a solid layer. It's actually got a super fancy name, phospholipid bilayer, which means it's got these little building block units. Imagine like the beads on a bracelet.
Yes.
And those beads on your bracelet are all stuck next to each other.
Yes.
If I was wearing a bracelet.
I'm imagining it.
Imagine the bracelet. So imagine all those beads stuck together, forming that layer around the outside of the cell. And you can have, like on Rachel's cell here, you can have other things sticking into that layer. You could have fancier beads on your bracelet and they'll all move about and they're all stuck together using those forces that hold small molecules together. So positive and negative forces, negative ones pushing things away, positive ones holding them together. It's kind of like when you have a mug on a coaster and you lift up your mug and the coaster sticks to it at the same time. Those kind of forces.
Science is everywhere.
Yeah.
Science is happening all the time, all around you.
When you're a scientist, you can't help but see it everywhere.
So cell membrane is holding all of that exciting stuff in. What is
Yes.
going on inside the cell?
There's so much happening inside a cell.
Tell me, quick.
But the plasma, I wanted to start with that plasma membrane because it's inside the cell as well. So it's that bag around the outside, but it also bags up a lot of these things in the middle that we call organelles. Okay. So in your cell, you have, I don't want to draw it, I'll do it with the non-drawing. But this kind of yellowy cream color that Rachel has stitched is what we call cytosol. So it's kind of like just clear jelly that's inside your cells. That clear jelly holds all these other bags of interesting things, which we call organelles. And probably the most important one, it's the biggest one in every single cell. You can probably tell what it is already from the picture. This big one in the center is your nucleus. And your nucleus has that plasma membrane around it again. So I like how you've done it in yellow here, and we've got yellow for the membrane as well.
Yes, yes.
So this inside your nucleus is your DNA.
Ooh.
Hopefully everyone's heard of DNA, or maybe genes, genetics, but your DNA is what codes everything inside your body. It's like a set of instructions for how your body works. And that DNA, it forms like a ladder. So you've got two sides that join up to each other and twist around. So imagine a ladder, but twisted. That's what DNA looks like. But that's so tiny, you could not see it under a microscope. So inside the nucleus, that DNA actually packs itself up. So imagine that ladder as a piece of string, and I start winding it up and scrunching it up together and forming it into maybe like a ball of yarn style. That's how we can see DNA in the microscope in the nucleus. It's all packed up together. And when your body needs to read part of it, it just unravels that one little part, reads the instructions from the DNA, and then ravels it back up again.
Fantastic.
It's very exciting.
Now, are we going to see some cross stitch?
Yes. So hopefully we're going to see some cross stitch. If we have, do we have any questions about cross stitch so far?
I forgot to mention, but if you have any questions for us, please put them in the chat. Behind the camera, we have Jenny doing all of our amazing tech work, and Jenny will read out your questions to us. So we did want to ask as well, if anyone watching, can you tell us about what level of cross stitch that you're at? Are you a complete beginner, never done this, you're a veteran cross stitcher, because we can adapt how we deliver this to you.
For instance,
Yeah.
I've never cross-stitched before. I'm the empty vesicle for you guys to fill
tell you
with
about
information.
Yes. So might go through just a 2-3 minute quick run through.
Yes,
Awesome.
jump in.
Awesome. So cross stitch is a type of embroidery. So there are many types of embroidery. This particular one is worked in little squares, and you do a cross over the square, hence the name cross stitch.
So you don't have to be very cross to do it.
No. Although
But you can.
stabbing something 1,000 times does help in terms of managing certain emotions. So cross stitch is usually worked on a fabric either called Ada, which has tiny little squares. I don't think you'll be able to see it. It's got like tiny little squares, and that can come in a variety of colours. So I've got one here that a friend of mine dyed.
Oh, wow.
That's gorgeous.
Yeah, that's called bushfire. So it's pretty
Is that
cool.
hand dyed?
Yes, it is. So, and then yeah, to work cross stitch, you need the fabric, you need some scissors, you need a cross stitch needle, and you need these fancy things called flosses, which you can get at Spotlight.
I've always called them embroidery thread.
Yeah, embroidery thread. Technically, there's a bit of a difference. I suppose
Okay.
embroidery thread sometimes is thicker, and use one strand, whereas with floss, it's sort of a little bit thinner, but either name works.
We're scientists. We're all about technical differences here.
So basically, you get something like this skein from Spotlight, and before you can use it, you need to get a little piece off. And if you're lucky, it worked for the video.
I love that.
Just
yes.
like the DNA
Ah.
inside the
nucleus.
inside the nucleus. I'm learning.
Yes. So then that gives us 6 strands, but we want 1. So in order to get a strand, you need to separate them out at the ends like this, and then you need to hold this really tight, and then pull.
Oh, you do that so beautifully.
Oh,
Mine always
practice.
just scrunches up.
Yeah, no, there's
There's a trick.
a technique. And then you can thread your needle and start stitching. So yeah, so I'm going to be stitching one of the little mitochondria as we go.
Fantastic.
the pattern.
Did you want to tell us about starting off?
Yes, I can tell you about starting off.
And then after you tell us about that, I might keep going on about the cell. Oh, do we have a question, Jenny?
We have a comment from Jess, who won one of our cross
Ooh,
stitch
yes.
kits.
Super.
Hopefully people can hear me. She said she's been cross stitching for 25 years since she was 10 years old, and that it's great for mindfulness. which we were gonna talk about later.
Ah,
Yeah, we're going
Yes.
to talk about that.
Thank you
later.
for that, Jess. Yes, it is really good for mindfulness. Do you want to tell us about that, Shasta?
Absolutely. One of the little things that I wanted to bring to the table, I'm really interested in how science is constantly impacting people. I talk about it a bit, how insects benefit people, but science of every agenda is also really useful and interesting to people. I think that you probably find it more interesting if you know how it affects you. And mindfulness, absolutely. The focus that comes from doing things with your hands. I've also got my insect puzzle on the table that I'm doing. The cross-stitching, crochet, knitting, the mindfulness that you get from that, it slows down your breathing, that lowers your heart rate, and it lowers your blood pressure, which gives you that kind of calm, thoughtful state. But that state that you create in the body can actually seems to influence your gene expression, that DNA in the nucleus. And so different parts of the cell are actually switched on, upregulated, or impacted by gene expression once you've entered that thoughtful, calm state of working with your hands. So some of the things that seem to be impacted by that is ATP production in the
Mitochondria.
in the mitochondria.
We will get to that.
We're
ATP
gonna-
is the energy of your cell.
We're going to get to mitochondria. I can see them there on the cell. For instance, your pancreas creates more insulin, which influences your blood sugar management, the amount of energy and the longevity of the energy that you have in your body, all from cross-stitching and telomeres, which are little extended parts on the end of your DNA.
I think of them like your little schemes.
Yes.
Schemes.
Skeins.
Say both. Telomeres are a little bit that sticks out the end there of your raveled up genes.
Yeah, and so they are the spare bits that you slowly lose as your DNA is replicated. And the longer they are, the longer your aging process is able to go on for. And so the calm state and the gene expression from crafting and other relaxing activities can actually make you live longer.
There you go.
We're going to live forever.
Awesome. So I'll show you how to start cross stitch.
Show us how to live forever.
Show us how to extend our
I don't
telomeres.
know if we're gonna. Can we do the
Can we do the
switch
zoomed in?
camera? Thanks, Tim. So here is my little piece of fabric, and I'm going to start. My mitochondrion is going to be about this shape here. So I'm going to start in this top corner and going to go through one of my little holes, which has been difficult. And then I'm going to pull my floss nearly all the way through. And then I'm going to go in the next hole. Sorry, there we go.
You don't tie a knot?
Don't tie a knot.
Ah.
And then so that's one half of the cross stitch, but I'm not going to complete that cross yet because I'm going to do a row of 4. So I'm going to go in the next hole and pull that through. And before I've pulled my floss through, sorry, there we go. So
Oh,
I've
is that how you make
secured
it neat at the back?
I've
I've always
secured
just left those hanging out everywhere, making a mess.
No. And as you finish the row, you can actually sew all of those under.
That's
and
fantastic.
And then go the other way around.
Ah.
Yeah.
So you don't do a cross like you mentioned.
No, well, it depends on the type of floss you're using. So these flosses are all the same colour. So you wouldn't do that. But if you use something called a variegated floss, which is the kind of floss that changes colour during the scheme, then in order to keep the colours together, you would finish each cross before you started the next cross.
That makes sense.
Yeah.
But in this case, you'll go along.
I'll
in a
finish
line.
that row and then I'll look at my pattern and I'll see where I have to go next.
Fantastic.
And so red, mitochondria,
Yes.
ATP.
ATP. Shall we talk about the mitochondria while you're stitching it?
Yes.
Keep stitching for the camera and I'll get excited about mitochondria. You might have
Wind you up
heard
then,
about it.
David.
You might have heard about it in class because every teacher calls it the powerhouse of the cell. All right, so what does that actually mean? I hate that sentence, by the way.
Yes.
It means that mitochondria create energy for your cells. So we eat food, that food gets absorbed into our body, and the mitochondria take that food in the form of glucose, which is a type of sugar, and they convert it to this really fancy molecule, adenosine triphosphate, or ATP.
Fantastic.
We just call it ATP for short. And that is actually the energy of your cells. So we don't use electricity like electronics do. ATP is like our body's own electricity, our power. And it's made in the mitochondria. So the mitochondria are possibly my favorite organelle though, because it's not as simple. They don't just make energy. There's actually a theory that mitochondria were their own cells in the past. They created energy for themselves. And then what happened in the past is our cells came and gobbled them up and started using them to create energy. So mitochondria have their own DNA inside them, and they have 2 membranes. They have their own membrane, which is the wiggly bit that you might see in the picture. And then they have a membrane around the outside that belongs to our cells, which is how we've swallowed them up. So you might see the little wiggly bits there and then around the outside. So you have DNA in your mitochondria that is different from the DNA in your nucleus.
Wow.
And you can actually use that for genetically tracking as well, because you share your mitochondrial DNA with your mother. Because when fertilization occurs, you have an egg, an ovum, and it is really large. it's for a cell. It's not for us, but for a cell, it's
cell, it's
quite
big.
large,
relatively
large. And it's full of mitochondria and all these other organelles and bits and bobs are in there. But the sperm cell doesn't have most of these things because it is a specialized, sleek cell that is designed to swim,
racing car cells.
racing car, swim fast and get into the egg. That's all it wants to do. So it doesn't have a lot of these extra organelles in the way.
Interesting.
So the mitochondria that you have in your body has all grown from that one egg cell that your mother had. So your DNA in your nucleus comes from the sperm and the egg, but the DNA in your mitochondria just comes from the mitochondria in the egg. So you can
Ooh.
track the matriarchal
Matriarchy.
lineage through mitochondrial DNA. And I think they're just so cool because we've just kind of absorbed them at some point in our past. It's almost like
Pac-Man style.
Yeah, Pac-Man style, where we're using these cells of our own to feed us, to give us energy.
So that was our mitochondria, not the powerhouse of the cell.
Ooh, we have another question, Jenny.
Questions.
We have a question from Facebook. Marie wants to know, regarding a thread, do you always use just one strand? And she also says it's great conversation about calm activities and DNA, and I recommend darning socks.
Darning
Ooh.
socks. That
Darning
is one
socks.
thing I haven't done yet.
I've never done that either. It kind of scares me a bit, to be honest.
So do I use one strand? What I do, because I am a bit lazy, there are different methods of doing it. I prefer using one strand and doubling it over in my needle so that my needle's always attached to my fabric, so I don't lose it. So that's my preference. And also at the moment I'm stitching on 18 count fabric, which has closer holes. So if I use 2 strands, that gives me good coverage of each of the squares as I stitch. If I was stitching on a bigger fabric, something like 14 count, I would actually use 3 strands to do my stitching.
I sometimes do freehand embroidery. So unlike the cross stitch, which is nice, neat ones, freehand, you go crazy, you just stitch whatever you want. And I'll alternate the number of threads I use for that. So something nice and small and fine, I'll use 1 or 2. And sometimes if I want a big chunky bit, I'll use, 3, 4.
Yeah.
Sometimes if I'm crazy, I'll use all 6.
Yeah, I crochet rather than knitting or cross stitching because I'm not very good at following instructions or rules.
This is where, if you're a designer, it's helpful. You create the rules.
Yes, yeah. I came to the conclusion partway through my craft obsession that crochet is effectively 3D printing.
I love crochet.
I
Yes,
love that technique.
with wool.
One bit at a time.
Yeah.
Yeah.
It's interesting that that creation process, and so there's kind of 2 sort of processes vying in your brain when you're doing craft. And so there's the repetitive motion, finding the puzzle piece, putting it down where you're kind of in flow, the 1,000 stabbing over and over again, where you know what your design is, and so you're following the instructions, and that creates that relaxation response that I talked about to begin with. But there's also this other element which Rachel is obviously deeply invested in, and as I was saying that I like, which is the creation part. It's that imagination of thinking up a thing that you haven't seen before. Rachel designs lots of different cross stitch patterns, and so she's using her brain to create pictures, and it's that creative process which does another really interesting thing inside of your body, which impacts your brain cells. Alana might explain to us in a minute. Neurons are different as well. They're a different kind of specialized cell, but they're the ones in your brain.
Yes.
You might have seen them. They've got these long dendrites.
They have axons and dendrites.
Yes.
The
Right.
dendrites are the fingery bits that stick out that take information in, and then the axons send the information out. So this cell is this is like we said, an emoji kind of made up cell that's got one of everything in it. But actually the cells in your body all specialize and do different things.
Yeah.
So like you said, neurons are brain cells,
Yes.
so some of many brain cells that we have, and they specialize.
And so, right, axon. So it is kind of like the arm is a decent metaphor for this, because when you use your brain, you exercise your brain cells, your neurons. And so when you're imagining, when you're creating a collage with pieces of paper, or when you're drawing a picture from your imagination, or following your heart's desire, creating your cross stitch on the fly, or designing the pattern, it's this imaginary process which actually causes different neurons of different lengths to fire, and the message has to travel at different speeds to arrive and create this imagined picture all at the same time. Rather than having a dolphin and a pineapple turn up at different times, you want a dolphin balancing a pineapple on its nose all at the same time. And that requires kind of different strength, different speed, and this exercises the neurons in your brain. And so thinking creatively, which is part of crafting, is also having this impact, this positive impact on your brain. Those neurons that fire together start to wire together.
I love it. Can we have that cross stitch?
Neurons that fire together.
And so
together.
that also creates neural networks. It makes your brain strong and robust. And so daydreaming, imagining, writing creative stories, thinking up pictures, animals, insects that never really existed before, and making them with your brain is doing incredible things for the cells inside of you as well.
You can exercise the cells inside your body, not just your muscles.
Dendrites.
I love it. Well, one of the things I did want to mention about this cell, I wanted to talk about how you actually get that information inside the nucleus and what it does, because we know that we have a nucleus here. We know it has our DNA in it.
String.
Yes, our DNA string.
Full of string, full of DNA.
Full of string in the center there. And we know it codes for instructions. But when I learned about this in university, it blew my mind, because the only thing that your DNA codes for, the only thing it's written down is proteins. That's all. It codes for proteins. So every single piece of information coming out of there then gets translated and added to until eventually you build an entire working body. I honestly don't even fully understand it. I think it's kind of a bit magical how it is. But
It's absolutely magical.
well, let's follow a journey of a piece of DNA to making a protein. So you start out in the nucleus, in the center of the cell, and you have to unravel a little bit of DNA to read it, to read those instructions. And there's a special other thing that does that called RNA. And RNA is exactly like DNA, that ladder, if you chopped it in half. So it's just got one side and all these chopped off ladder steps sticking out. So RNA comes along, and what it does is it unzips your DNA, splits the ladder in half. It reads the rungs of the ladder. It matches up. So it's writing down the instructions. And we call this messenger RNA because it's writing down instructions. It's a messenger. And what that does, once it's read your DNA, it'll zip it back up again, and it will come, hopefully, I hope it zips back up again, and it comes out of the nucleus. So Rachel on her nucleus here has made several holes in it, because it's not a solid bag holding your DNA. It's very holy indeed.
It's like the gaps in the beads.
Yes, yeah, gaps in the beads of a bracelet.
Yeah.
You can send that messenger RNA. I lost my bug. You can send that messenger RNA that's read the instructions out through the pores in the nuclear membrane or the nuclear envelope. And what happens then is someone's got to read those instructions.
Who's going to read it?
Who's going to read it? That we have a ribosome. So if you ever look at a ribosome under a microscope, you might see kind of like on this, just these tiny dots. So a lot of people don't learn anything more than that in school. They just learn, oh, ribosome's a dot, it's tiny. But they're not. They are so exciting. Possibly my second favorite organelle after mitochondria, because they're not just a tiny dot. They are protein builders, and they have two halves. They have a bottom half and a top half. And Rachel has even made some big purple ones. So but we have tiny little ones, how they look under a microscope, and big cartoonish purple ones there with two halves.
Emoji ribosome.
Emoji ribosomes. And this messenger RNA comes out and slips between the two halves of that ribosome, and the ribosome reads it. It reads that message. and it pulls up another type of RNA. So that half of a ladder is called transfer RNA or tRNA. And that tRNA transfers an acid, a tiny little amino acid. It has a little dot of a molecule attached to it. Every single different transfer RNA is for a different amino acid. And ribosome reads the message and it says, I need this acid because the message tells me so. And it pulls one in and links it together in a chain. And then it goes to the next bit along on the message and does it again. Pulls it in between the two halves of the ribosome, attaches that amino acid and moves on to the next one. So by that, you get to, like a bracelet again, you're putting beads on a string and you're building up a long string of amino acids.
Just following the pattern.
Just following the pattern, exactly
With
like
the
cross-stitching.
RNA is copied from the DNA.
Yes.
I
So
can
we
see
have
that.
to go through a couple of steps of copying, but what we end up with is this string of amino acids which forms a protein. So you probably know about proteins from meats or tofu, or because muscles are made up of protein because most of our cells are made up of proteins, among other things. So that excites me a lot because it blew my mind in university when I was told that DNA that codes for everything in your body just makes proteins.
Everything is protein.
Everything's protein. Not exactly, but what happens, not everything is proteins. What happens next is you sometimes then package those proteins. So you take that string of beads and you might fold it up somehow, or you might add other things onto it. You might add lipids, fats, or cholesterols, or other molecules to it. And that's how we fold it up and make it into the things that our body is made up of. Right. So one of those things happens in the endoplasmic reticulum.
No.
No?
No, I'm overwhelmed.
You're overwhelmed? Thank you for stopping when you're overwhelmed.
I want to look at more cross stitch.
Right. So at the moment, cross stitch is a slow craft. So this is our progress.
How long? I want to ask a question from Facebook. How long did the whole cell, or maybe the small one, take to cross stitch?
Yeah. So so this little cell, this one here, this one has about 3,000 stitches. The little plant cell, this one here, that has about 4,000 stitches. This one here, 12,000 stitches.
I love that you counted.
Roughly. And it took a little while to stitch, but also to plan it so that everything worked in terms of making sure that the proteins connected and the little vesicle connected and all of that sort of stuff. So,
But people can cash in on, capitalize on you having already designed
I've already
this
designed it.
pattern, whether they're stitching along today or whether they might want to pick it up now that they've seen how beautiful it is. Your pattern exists somewhere, doesn't it, that they can go and get?
Yes. So I've got the pattern for this minus the fancy polymer elements, just because the size of those would depend on the count of fabric that you were using.
So it would look more like that one.
Yeah. So I've got the big one, and then I've got the small one, and then a plant cell. Yeah. So yeah.
How long would it take without the patterning, do you reckon, for someone to do that? It'll just depend on how much they cross stitch.
Probably a little bit. Like I think for a new person, probably you're looking at 2 weeks of evenings maybe
That's not
to
too
finish
bad.
that.
Do we have a question, Jenny?
We do have a question, and this one's probably for Shasta. Has there been any research that you know of showing if crafting can help people with memory loss or keep elderly people's minds sharp? That question is from Deborah.
Deborah, absolutely. Whether you've thought of that off the top of your head, you're spot on the money, because there's lots of research that actually shows that exact thing. I'm sitting here looking at my little list of craft and health, and so the mood boosting kind of attribute of crafting and getting into that relaxation zone, but also the it's kind of referred to, I've seen it as ambidextrous brain. And so your brain has a left and right hemisphere, and that links to the left and the right side of your body, but in opposite.
So your right hand is controlled by the left side of your brain and vice versa.
Well, that works. If you're shaking your left hand, you've only got your right hemisphere. You can only point to your right hemisphere. That's a good way of remembering it. And much like my axon, my strong axon, I'm right-handed. A lot of people are. And so my left hemisphere gets all of that work that my right hand does. I write and I use my computer mouse, things like that. And so my left hemisphere gets all of this work, all of those neurons knit together, and they make a really strong network. Whereas my right hemisphere, my left hand, doesn't get as much exercise, doesn't get as much work. And so it's less strong, less healthy. And as I age and some of my telomeres get worn down, the little DNA extra bits, all of my body, including my brain, starts to age and get less strong, less robust. But you can create an ambidextrous brain, which means a left-handed and a right-handed brain, by bringing in your weaker hand, your weaker side. Craft does that because, or any activity, any of that calming activities that uses both of your hands. So if you were doing a puzzle, if you happen to like doing puzzles, you can tell yourself to use your left hand for the next minute to pick up pieces and put them in. It's a little bit harder. You're probably less coordinated with your offhand. And so you can tell yourself that the struggle is worth it because you are creating an ambidextrous brain. Some of the things that I like to do, I move my computer mouse to the other side of my desk just for a few weeks or a few months at a time. I have challenged myself to brushing my teeth with my left hand, which is very unpleasant and is best done in private.
You need to try it. I've tried it and I thought it would be fine, and it was not fine.
Yeah. But some of the human conditions that can be improved by the calming effect or by the neural improvement in humans, things like pain can distract you from pain. It can distract you from anxiety. Conditions like PTSD or depression, but it can also help with the aging of the brain or with any kind of brain injury, acquired or aging related, which can be sort of overcome or pushed back against by developing those neural networks. Other things that I thought of or that have been mentioned, practicing an instrument, like the calm, quiet time, not the stage anxiety, not the stressful part, or practicing your sport. I thought maybe hitting a tennis ball up against the wall, but then now you know that you also have to practice with your offside or with your offhand. So again, it's the left hand and the right handed activity that really brings all of that scientific knowledge and all of that physical, emotional benefit together.
But even if you are not using both hands, it's still exercising your brain and doing all those things to do something like craft or learning, pretty much thinking in general. One of the things that blew my mind when I started learning about the brain was that just sitting there doing absolutely nothing all day, your brain would use up 20% of your body's energy. So it's tiny compared to the rest of our body, but 20% of it just gets used just to exist.
Runs hot.
It runs hot. It does. So when you are thinking and doing things, you might have noticed during exam time, you get hungrier because you're actually using a lot of energy, and it's really good exercise.
I thought that was just because chocolate makes me think that I'm having a nicer time.
Bit of column A, bit of column B.
Have we gone over everything that is on the cell?
No, we have
pattern?
so many parts to talk about. Chop. This, I don't want to speed through too much, but well, the next part in our story was the fancy words I've mentioned once, the endoplasmic reticulum. So again, this is a membrane. This is another phospholipid bilayer. You can see on Rachel's cell here, these blue-gray parts are the inside of the endoplasmic reticulum, and these yellow parts of that membrane outside. And Rachel has stitched them in yellow, I'm guessing, because it's the same color as the nuclear membrane here.
Because it's a continuation of the nuclear membrane.
So your nucleus is not just a bag with holes in it, it's a bag with holes in it and bits that stick out, little fingery bits. I'm great at explanations. It's very complex, but it's beautiful. And those little fingery bits that stick out of the nuclear envelope there are the endoplasmic reticulum. So what happens is ribosomes, those purple ones there, those dots, they can actually attach themselves to this endoplasmic reticulum, like docking on maybe think of like a space station with all the ribosomes being the ships. Endoplasmic reticulum's a space station. They all go dock in their little holes on there.
I like this.
And they will make proteins partially floating around in your cell, partially attached to the endoplasmic reticulum. And that endoplasmic reticulum will help package the protein. They'll help add extra bits to it, fold it up in certain ways so that it can do its job. You can have two types of endoplasmic reticulum. There is smooth and there is rough. And the only difference is rough has ribosomes attached to it. So it looks rough under a microscope. It's got lots of dots. And smooth doesn't have ribosomes attached. And the smooth portion does a lot of lipids, so fat packaging and making.
Smooth like butter.
Smooth like butter. Butter is fat.
Yes.
Lipid.
I know that, Alana. I'm very smart.
The next bit in our story, I would say, would be this orange bit here, which you would call the Golgi body or the Golgi apparatus, or just the Golgi if you feel like it.
if you're on first name terms.
Yeah, first name terms with it. Golgi thingy. So what happens next is these ribosomes have read the RNA, they've made proteins, they've maybe docked in the endoplasmic reticulum and packaged the proteins a certain way. Then the endoplasmic reticulum will send those half done, half folded proteins out to the Golgi body. And this one is kind of like stacked on top of each other. I kind of think of it, sometimes I have a whole lot of shopping bags and you'll just shove them one on top of the other.
Oh, yeah, everyone's got that drawer.
Everyone's got that. So the Golgi body's like that. It's like bags, shopping bags full of protein shoved on top of each other. And all the protein comes in one side. So that's me adding my extra shopping bags. And then I'll pull the old shopping bags out the bottom. So it's got one side where the protein comes in, and then it gets packaged and transformed and folded and cool bits are added. And then it comes out the other side of the Golgi body, hopefully nicely finished protein that can do a job, that can build a cell or tell something else in your body to do something. It could affect other molecules, and they go out and they'll do their job. It can send it out in these little things though called vesicles. So these vesicles, I feel like I've always thought of them as kind of vehicles. It's close enough. Yeah, it works. They're just bags full of something, in this case protein. And these vesicles, I think you've got some on here, don't you?
I've got those ones there.
You've got these little orange ones.
There's some on the small one we can hold up.
Yep. So
Are you able to swap to the other camera, Jenny?
these little.
So these
little ones.
vesicles are full of something. And because they are a plasma membrane, just they're wrapped up in a membrane. That's the outside is a plasma membrane, same as the nuclear envelope, the endoplasmic reticulum, the outside of mitochondria. They can just join on to the outside and they can just pop open and release their contents outside the cell. So that's how you get something out of the cell without ever having to open up the membrane. You just join an extra bit of membrane to it and it pops out.
Spaceship airlock.
Yeah.
Docks on the inside, opens the airlock.
I'm liking these analogies.
I like it.
I
That's
like
good.
the spaceship analogy.
You could do it the other way as well. I see on your cell that you have done one going in. So if you want to take something into the cell without opening up the plasma membrane, you can just fold over a piece of the membrane. And once it's folded over and the ends touch each other, it will just pop off. And it will have a bit of a membrane bag inside the cell now. So that's how we get things in and out. What else are we missing from this cell? There are so many bits in here.
Yes, many little.
Many
bits.
little bits. What about, we have lysosomes, don't we? These are
Yes.
our green dots here.
Mhm.
So lysosomes are, again, the plasma membrane around the outside. They're just a bag full of something, but these somethings are specifically for digesting.
Yeah.
So you might ask, what could the inside of a cell digest?
I do want to ask,
Yeah.
what could the inside of a cell digest?
It's not food. itself a lot of the time.
I feel like this is a dangerous precedent.
So sometimes something goes wrong in your cell. One of your organelles might not be good anymore, needs to be gotten rid of, recycled, and a lysosome will come and gobble it up and digest it. Or maybe the whole cell is reached the end of its life. It's going to die. So the lysosomes will digest the cell. Or maybe you've got an intruder into the cell. Something came in,
Ooh.
something managed to get through the plasma membrane. The lysosomes can surround it and digest it.
What kind of things? I've got lots of words sleeping through my mind, like bacteria or viruses and things, but
Yes, bacteria, viruses, not sure, bacteria and viruses. I mean, you could get physical things in as well. Something could push through the cell, but often then it will just rupture the cell and the whole cell will break.
That would be more of a cell death situation rather than a lysosome situation.
Yes, exactly.
Cool.
What else have we got? I've got a picture here that Rachel's
We're
labeled.
coming to the end of our time, aren't we? This means that people at home, this is your opportunity to ask biological questions, cross-stitch questions before you're on your own again. Aside from the fact that
Yes.
all of us are contactable and obviously have more that we'd be willing to say about
Oh,
science.
it's hard to make me shut up. Well, before we finish, if we have, please ask questions in the chat and we'll stop and answer them. But we have some other little bits. So down in the cell, what bits? Other bits. There's so many bits. What about that one? That was quite big.
Yes. So that one there is a vacuole. So
We're looking at the blue
we're looking
circular
at the
bit.
blue circular bit. Yeah, so that's like a fluid-filled space inside the cell. They can be used for storing different things. So, for example, your fat cells in your adipose tissue will have large vacuoles filled with fats, and then that's how they store them.
Plant cells as well have a special kind of
They do.
vacuole in them.
Plant cells have a very large vacuole in them that they use to store water. So it keeps the cell hydrated and keeps it pushed against the cell wall, because plant cells, unlike animal cells, have a cell wall.
Yes, I love this. So animal cells just have that membrane around the
Mhm.
outside. It's just like a bag. But the plant cells have a cell wall as well.
Yes.
So it's like putting a bag inside a cardboard box.
Yes.
So they've got a membrane as well, but it's
Yes.
inside the plant wall. So that vacuole puts pressure on the bag. Think of the bag really full of water and it presses up against the cell wall.
Mm.
And if you don't have much water in your vacuole, the bag shrinks a bit, but the cell wall stays the same,
This is how your plants are able to wilt without actually just
turning to mush.
deflating.
Yes. Yes.
But when you freeze your vegetables and then thaw them out,
Yes.
the vacuole, because water expands, has actually breached the cell wall. So all of that cardboard box structure is
all
broken. And that's why your defrosted vegetables collapse. They do
Some
collapse
of them, yeah.
rather than just wilting.
Yes.
Yes.
I love that. That's why you just can't put some, you can put some vegetables in the freezer, okay? Like peas and corn, and others you just cannot.
Science everywhere.
So a lot of these extra bits here are not organelles in their own accord. They're not these little bags of membrane bound things. Did you want to talk us through some of these little swirls?
Curly bits.
Curly
Little
bits?
curly bits. Oh, weird. Well, I found this wire from a craft shop somewhere or op shop somewhere, so I had to use it. I thought it was funky. So a lot of these little bits that I put here are elements.
Jenny, are you
called-
able to swap to our zoomed in camera? Sorry.
Sorry,
We realized we were
I'm-
talking about
I'm
things.
talking about things you can't see.
Yes.
Literally,
There we
this
go, these
entire
little-
thing has been a conversation about things that people can't see.
That's
True.
Technically.
True. These
So
little
yes,
bits.
so these little tiny bits here, they're microfilaments and microtubules. So they're
Ooh.
elements of a cytoskeleton of the cell.
I love that word.
Yeah, the cytoskeleton.
Not
So-
only do you have a skeleton inside you, you have a skeleton inside your skeleton.
So they're basically the parts of the cell that hold everything in position, and they can help in terms of movement of things from one particular part of the cell to the other as well.
Like climbing over a scaffold.
Sort of, yeah.
Yes. So your cell has all these microtubules, microfilaments, and intermediate filaments that
Yep.
all together form like a spider web inside the cell. And that spider web is the cell's own skeleton.
Yep.
and how it moves things around. They don't just float around everywhere. They actually get dragged along those filaments like they're railroads. So you can drag things around your cell. And that's also how cells separate when they, when you have cell division, when a cell clones itself and splits into two of the same cell, it will use those cytoskeletal bits to pull everything apart, like little fingers.
Yep.
And you've also made part of that,
Yes.
the centrosome.
Yep. So you've got this is the centrosome, you can see, and these are centrioles, which are special sort of cytoskeletal components. They're microtubules that are in a special grouping at right angles to each other. And they're really important for cell division. So they direct some things go to one part of the cell, like all of the half of each chromosome goes to one part and half goes to the other.
So they end up, those little centrosomes will split up and end up one on either side of the two cells. And it's almost like they're the thing pulling the two cells apart.
Yes.
With their skeletons. Yes.
Red team and blue team, everyone go and stand at opposite ends of the gym. And then the gym is going to split in half and turn into two gyms. Okay, this metaphor needs work.
Not the best metaphor we've done today, but probably not the worst.
We're so close to the end. Can we see how your active
Yes, my active stitching. on my-
ribosome?
Right, mitochondria.
Yes. So this is my progress so far.
Up a bit, up a bit. There we go. There we go.
Sorry, not very good with these cues. So yeah, so it probably will take, maybe 20 minutes to do a whole mitochondrion as a rough indication of time. So
I love it though. You've got the membrane around the outside. Oh, point the pen away so I don't draw on it. You've got the dark red for the membrane on the inside, and then that light red for the bits inside the mitochondria's own.
Yep.
What's the word? Cytosol.
yeah. Oh, what are they called? Cristae?
Yes, the
complex
little
folds.
The little folds inside the mitochondria are called cristae. Look, we love naming things as scientists. Everything inside the cell has a good 5-10 names.
So
it is gorgeous though. I've been super excited to be able to come here today and see this craft and just spout my science, talk about cells.
Absolutely. How crafting has all of those positive impacts. How crafting a cell can actually influence your actual cells.
This is so meta.
I found it incredibly detailed and exciting. I mean, that's why there's so many names for things, because there are so many things inside your things being influenced by the things that we do. Probably don't quote that. It'd be a very long cross stitch. So we're hoping that some people have been actually cross-stitching along at home. We've a number of competition winners who actually received all of the equipment necessary and the design that you designed with your brain, making your brain stronger. Are we anticipating seeing those photos somewhere? People can send them in and share with us on the Facebook page where the event was
please share them with us. That would be
located.
amazing.
Yeah. And
what is your company?
So my business is called Fuzzy Fox Designs. So yeah, it's on Etsy, ETSY.
It's on Facebook as well, isn't
it?
It
Is.
So I like
it's
the
on
page.
Facebook and Instagram.
So Rachel, as Fuzzy Fox Designs, put together these kits for some of the winners. And you can see these cells and other work that Rachel has done on the Facebook page and on Etsy.
Yes.
So we've got other events too. You might have seen me. I've been attempting to helpfully point at things while I am also following along. You can follow along at home. Well, I've been following along in the studio, but what I'm using, if you haven't seen one of these yet, is our National Science Week Science at Home brochure.
You can get that for free online as well if you can't get your hands on a hard copy. And there's many other events in there.
Yes, chock full of
creative events as well.
Other things. We've got a beer.
Yes, we have
taste along.
Science of beer. We have the science of other foods like sourdough.
Taste along at home.
Sourdough and other fermented foods. There's a kitchen science page in here. We have Tastrofest is going on for most of Science Week. We are Young Tassie scientists as well.
Yes.
Young Tassie scientists normally go around schools during Science Week, but this year we're entirely online. We're doing virtual visits to classrooms and we have videos on YouTube. watch
Yes.
our videos.
Which means you don't have to be enrolled in a primary school this year to have access to Young Tassie Scientists. Anyone can come along. See
Anyone, please.
my bug videos, Alana's
Yes.
brain videos.
We have introductory videos for all, I think, 18 or so scientists. There's a lot. Look, Science Week this year is it's fantastic every year, but National Science Week Tasmania this year has so many events. We really appreciate you coming along to this one, cross-stitching along with us maybe, or just learning a bit about cells or your brain. We have had fun, and we hope that you've had fun too.
See ya.
Bye.